Welcome, AP Bio students, to your review of the cell cycle and meiosis. This is chapters 12 and 13 in Campbell Biology. I'd suggest you do the reading if you hadn't done so already. You have a test coming up shortly here, and this is a quick little review of information that I know will be on that test as well as on your AP exam. So let's pay attention and we'll go through the highlights of the chapters here. We'll start with mitosis versus meiosis. You might see a question in your near future that has something to do with comparing and contrasting mitosis and meiosis. They're two very different processes. Mitosis, first of all, deals with just duplicating a nucleus, whereas meiosis, you're actually creating, it's how gametes are actually created. So the end product of meiosis is 4 genetically unique cells, whereas the end product of mitosis is just 2 duplicated nuclei. So let's start with mitosis, and I'll go through some bullet points of unique features of mitosis. Like I just said, it's how copies of DNA move to opposite poles of a cell. You start with a diploid cell. One diploid cell enters mitosis. In biology shorthand, we would call that a 2N cell. It's got 46 chromosomes is what that means. Twice the haploid number for humans, which is 23. It ends with a cell that with 2 diploid daughter cells, each being 2N. Each are diploid. Now they have unduplicated chromosomes in those daughter cells, so you would have gone from X-shaped chromosomes to stick-shaped chromosomes, but you still have 46 chromosomes when you're finished. So the important take-home point about mitosis is you started with one cell that had 46 chromosomes. You ended with 2 cells that both have 46 chromosomes. Usually mitosis is followed by cytokinesis, which is the actual physical splitting of the cell that contains our chromosomes. Compare that with meiosis. This is how gametes are made. Gametes is a science word for sex cells, the sperm cells and the egg cells. You start with one normal diploid cell, just like we did with mitosis. But in the end, what we're going to produce is 4 haploid cells. So in a human, that would mean we went from one normal diploid cell that had 46 duplicated or X-shaped chromosomes, and we ended with 4 haploid cells that have 23 stick-shaped chromosomes. And each of those 4 resulting cells is going to be genetically unique. It's non-cyclical, whereas mitosis, you could have taken either daughter cell and allowed it to go through the cell cycle, and that cell would be capable of doing mitosis again. Meiosis doesn't work that way. Once meiosis has been done to a cell, that resulting gamete is either going to be used for sexual reproduction or is just never going to be used. Bummer. Oh, let's see. What we're looking at here is, on the bottom, we have a video of the stages of mitosis occurring. And the stages of mitosis are listed over here on the left: prophase, metaphase, anaphase, telophase. I also included up at the top here a static picture of a microscope slide. Right now the cell is in prophase, the cell at the bottom that's under in the movie clip. And now we're entering metaphase. See how the chromosomes are lining up across the middle of the cell. I always remember that metaphase means they're lining up in the middle. Right about, wait for it, right about, now we've entered anaphase. And finally, as the 2 new nuclei form on opposite sides of the cell, we have telophase, and then followed by cytokinesis, and you can see the 2 cell membranes are dividing at this point in the video. So that's a quick little rundown of the stages of mitosis. I would make sure that I know those. The easy way to remember, of course, is PMAT. PMAT, right? Prophase, metaphase, anaphase, telophase. The cell cycle, which mitosis is a part of the cell cycle. This is the cell cycle, and you see a lot of the CDKs around the edge of this cell cycle. You should remember that CDK stands for cyclin-dependent kinases. These are proteins that regulate progression through the cell cycle. Think of cyclin-dependent kinases. They're a signal transduction pathway that's made up of proteins. And these activated signal transduction pathways allow the cell to proceed to the next step in the cell cycle. So the cell cycle contains several stages. There's 2 growth stages. The cell cycle starts in the G1 phase. where the cell grows and carries out its daily activities. G1 is followed by the S phase, and which is where DNA is synthesized, which is then followed by the G2 phase, which is another growth phase for the cell. So the S phase is where we actually replicate our DNA. We go from stick-shaped chromosomes to X-shaped chromosomes, because you know that an X-shaped chromosome is really two sister chromatids that are identical. Cell division occurs at the end of the M phase. M phase includes both mitosis and cytokinesis. And it's cyclical, so once we have those two daughter cells at the end of cell division, they would re-enter G1 phase and continue around the cell cycle. And that's how you went from being one cell big to several trillion cells big, is just by taking many, many, many trips around this cell cycle. All those trips around the cell cycle are regulated, the speed at which you go through them are regulated by these CDKs, these cyclin-dependent kinases. I would make sure that I knew quite a bit about those. Finally, a little bit about the chromosomes. We talked a lot about stick-shaped and X-shaped chromosomes in class, but you should be familiar with the technical terminology. We have sister chromatids here held together at centromere. It's important that you understand for each and every chromosome, numbers 1 through 23, you got one chromosome from mom and one from dad. This picture here, I'm trying to show you that the two sister chromatids are identical DNA in one chromosome, whereas the chromosome on the left and the chromosome on the right, the one that you got from your dad and the one you got from your mom, those would be homologous chromosome pairs. They would have the same genes, but they had potentially different alleles. And I would understand how all that business hangs together. So there you have it in writing right there. Homologous chromosomes, they have the same genes. So all chromosome number 21s that are human chromosome number 21s, they all have the same genes, but they have potentially different alleles for those genes. So for example, if on chromosome number 21 I found the gene for hair color, okay, that would mean that every chromosome number 21 on Earth has genes for hair color on it. But that means that on some chromosome number 21s we would have the allele for brown hair, and on some chromosome 21s we would have the allele for blonde hair. So maybe on the one on the left you had the allele for brown hair genes, and the one on the right you had the alleles for blonde hair genes. Sister chromatids, however, the left and the right side of the X-shaped chromosome, they're going to have the same genes and the same alleles, assuming a crossing over event hasn't happened yet. Speaking of crossing over events, I would make sure I understood that at metaphase 1 of meiosis, that crossing over events do occur, which means that you end up with homologous, you end up with sister chromatids that are not necessarily carrying exactly the same genes. I would read about crossing over events and make sure you understand how you can get sister chromatids that do not have identical. Well, they have to have identical genes still, but they don't necessarily have to have identical alleles for those genes. And if you don't know what a crossing over event is, you're going to want to know by the time your test rolls around. Oh, finally, the end of this review video. Yeah, Physics Cat, you're very funny, very funny. Notice it's a Campbell biology book, though. Physicists like to make fun of biologists because they think it's a soft science. They can talk when they cure cancer. Anyway, that's everything I think that I needed to hit on as far as cell cycle and meiosis goes. Make sure that you read the chapters in the book and be ready for your test coming up shortly here. Good luck studying. Have a good night.